Cancer is one of the leading reasons of death in humans, with the mortality rate being close to 11 to 25%. Medical professionals and scientists alike have been searching for preventative measures and treatments of varying cancers for many decades now. A recent finding regarding elephants' cancer mortality rate has meant development in cancer research. Despite their large size theoretically meaning elephants should be at a higher risk of cancer, the species has a surprisingly low mortality rate, 5%. This lack of correlation between size of organism and cancer risk is known as Peto's paradox. Two genes appear to be at the center of this breakthrough, the TP53 gene and the LIF6 gene. While humans only have one copy of this TP53 gene, elephants have at least 20 copies, which could be the reason for this low mortality cancer rate. Elephants have more than 11 extra copies of the LIF6 gene as well. Researchers work into not only researching why the species has these extra copies of the genes than humans, but also how to replicate and transfer this into human medication would greatly assist the race to prevent and lower the cancer mortality rate.
Peto's paradox focused on the observation that there was no correlation between the increased number of cells in an organism and the incidence of cancer. Due to the high rate of mitosis, it was expected that there would be a strong link between increased body size and increased cancer burden, therefore linking larger body size and increased cancer mortality rates. This was shown to not be the case in the world's largest land mammal, the elephant. Due to elephants' extremely large body mass, it would be expected that they had a higher incidence of cancer compared to smaller mammals, such as humans. However, it was shown that at a species level, the incidence of cancer did not correlate with the number of cells in an organism. The following information presented in the video will discuss how and why Peto's paradox was incorrect. However, there are many simpler and more straightforward reasons for elephants' indisposition to cancer. The diet of African elephants consists mainly of greens, and they are considered a herbivore species. In recent studies, there have been immense investigation into how diet can affect the development and incidence of cancer. It has been suggested that 30 to 40% of cancers can be prevented with a healthy diet. A large portion of this can be attributed to a diet consisting of vegetables, removing red meat from the diet. This causes an imbalance in omega-3 and omega-6 fats, and a study conducted by Devisi et al. Suggested that a diet consisting of vegetables and fruit is linked to a lower risk of cancer. While this has not been studied in the elephant species, it could be linked to their lower incidence of cancer. A further reason is linked to the small genetic pool of African elephant species. As previously discussed, cancer is caused by mutations in the DNA of an organism. Thus, a smaller genetic pool would be linked to less genetic mutations and therefore less complicated cancers. Due to urbanization in the African continent, as well as the large number of elephants in captivity, the genetic pool for African elephants is restricted and small. This could play a large role in the low cancer incidence and mortality rate in the species. This was not addressed in the information presented and could also be linked to elephants' lower incidence of cancer.
TP53 is a gene that plays a key role in controlling cell division and cell death. It does this through the production of a specific protein which is found inside the nucleus of cells. Mutations within the gene may also cause cancer cells to grow and spread in the body. The gene plays a critical role in suppression of tumors by inhibiting the division and growth of cells with damaged DNA. It is important to note that over half of all cancers that occur are caused due to a missing or damaged TP53 gene. The gene codes for a protein that is pivotal in the body's natural defense mechanism against tumor cells and the suppression of tumor formation. The gene is also capable of conducting methods such as apoptosis, which causes the damaged cell to terminate itself and prevent further reproduction. All aspects of cancer, such as its aggressiveness, response to treatment, and its ability to spread to other sites in the body, are related to improper function of the TP53 gene. If a person inherits only one functional copy of the gene from their parents, they are generally predisposed to cancer and usually develop several independent tumors in a variety of tissues in early adulthood. Studies suggest that the elephant genome consists of 19 times more TP53 genes than that of the average human. This makes their cells significantly more sensitive to damaged DNA and quicker to engage in cellular apoptosis. The human TP53 protein has a structure that consists of 393 amino acids and 4 major functional domains. At the N-terminus is a transcriptional activation domain, and within the central part of the gene is the sequence-specific DNA binding domain. The C-terminal contains a tetramerization domain and a regulatory domain.
Another important aspect of elephant DNA that plays an important role in cancer resistance is a particular copy of the leukemia inhibitory factor gene, or LIF, which produces the LIF protein. Part of the interleukin-6 class, it is characterized by 4 anti-parallel alpha helices joined together by the secondary structure of the protein. The helical shape allows for a large surface area to bind to different receptors. The gene is common amongst mammals and has a variety of functions, but is mostly associated with inhibiting variety during cell growth. The majority of mammals only have a single copy, but elephants and some of their close relatives have as many as 11 of them. All but one of these duplicates are pseudogenes, sequences in DNA that have been mutated over time to an inactive state. However, a study done by Chicago University found that one duplicate of LIF in elephants, LIF6, was not as useless as the others and played a role in cell apoptosis just like TP53. This suggests that LIF6 had been mutated to become inactive and then mutated again, given a new purpose. We could say it was resurrected, aptly labeling LIF6 as a zombie gene. So how does this zombie gene make elephants so much more resistant to cancer than humans? In short, elephants are much more sensitive to DNA damage than we are, so apoptosis occurs much more frequently in their bodies. In fact, LIF6 does not even need DNA damage to induce programmed cell death. Of course, it's not an incredibly common occurrence and scientists are not even sure if it occurs in nature, but it does demonstrate how much more potent elephant apoptosis is compared to humans. It isn't entirely clear how LIF6 induces cell death, and so is something that requires future research. However, what is clear is the relationship that LIF6 has with TP53. On LIF6 there's a binding site for the p53 protein and scientists have determined that there's a reason for it. Though LIF6 doesn't need the p53 protein to function, p53 is what makes LIF6 much more responsive to DNA damage. In other words, TP53 upregulates LIF6. LIF6 is structurally very similar to LIFT, an isoform of the LIF protein. LIFT even has binding sites for p53, which leaves us wondering, could our bodies have the tools and materials to replicate what LIF6 does for elephants?
Hopefully, researchers' work into elephants and why their mortality rate is so surprisingly low can ultimately assist medical professionals in overcoming cancer one day. Elephants' extra copies of the two genes TP53 and LIF6, which humans don't have as many of, may be the clue researchers and medical professionals have been looking for. So we will just have to wait and see if the human race can overcome cancer anytime soon.